Good morning, children. Today we will begin with a new chapter, Photosynthesis in Higher Plants. The combination of carbon dioxide and water by the plant to form carbohydrates is termed carbon assimilation. And since the chemical process involved requires light as a means of supplying energy, this process is usually known as photosynthesis. Photo means light and synthesis means manufacture or combination. This process is almost always accompanied by the evolution of oxygen, and it takes place in the presence of chlorophyll. So two most important components here for the preparation of food by the plants are chlorophyll and sunlight. The entire process can be represented by the following equation. CO2 plus H2O in the presence of light and chlorophyll form glucose and evolve oxygen. So, photosynthesis is the only biological process on earth that releases oxygen. Okay. Now, studies by Rubin and Kamen 1941 indicated that oxygen molecules that are released during this process are contributed by water. So, oxygen is not coming from the CO2, it's coming from H2O, water, which means that 6 molecules of water are insufficient for releasing 6 molecules of oxygen. Therefore, you can modify the above equation as 6 CO2 plus 12 H2O in the presence of light or chlorophyll gives C6H12O6 glucose plus 6 O2, 6 molecules of oxygen plus 6 H2O. Historical perspective and contributions of some scientists. You'll have to learn the contributions of these scientists. Joseph Priestley, 1733 to 1804, performed a series of experiments that proved that green plants purify air and produce oxygen. Jan Ingenhousz, 1730 to 1799 showed that sunlight is essential to the green plants for photosynthesis. Julius von Sachs proved that green plants produce glucose by the process of photosynthesis, and he also showed that chlorophyll is located in chloroplasts. T.W. Engelmann described the first action spectrum of photosynthesis, and he proved that maximum photosynthesis occurs in blue and red light of the spectrum. Cornelius van Niel demonstrated that photosynthesis is a redox reaction in which oxygen comes from water. Now we are going to study about the site of photosynthesis. Now here there is the electron microscopic structure of a chloroplast, and in a cell photosynthesis takes place in chloroplasts. It is a green plastid. Chloroplast is a double membrane bound organelle. So here you can see that there is an outer membrane and an inner membrane. Each membrane is 60 angstrom units thick. The space between the outer and inner membrane is called as periplastidial space, and it is 100 to 300 angstrom units. The inner space is filled with a granular and transparent fluid known as stroma or matrix. The stroma contains fat globules, starch grains, osmophilic granules, pyrenoids and enzymes of the dark reaction. The matrix also contains RNA, DNA and 708 ribosomes. And this was discovered by Robinson and his collaborators in 1962. In the stroma, a characteristic system of lamellae is present made up of unit membrane bound structures called thylakoids, 100 to 300 angstrom unit wide, and these thylakoids are stacked over one another. One such stack is called as a granum. A granum may comprise about 50 to 100 thylakoids. In a chloroplast, usually 40 to 60 grana are found. The grana are interconnected by intergrana lamellae, also called stroma lamellae or frets. On the inner surface of thylakoid membranes, there are particles of 185 angstrom unit length, 150 angstrom unit width and 100 angstrom unit thickness, and are called quantosomes, discovered by Park and Wiggins. And these are smaller photosynthetic units. What are small photosynthetic units? Quantosomes. And they are capable of carrying out photochemical reaction. Photosynthetic pigments. Now, solar energy is trapped by pigments that are present in the membranes of the chloroplast, and these pigments are of three main types: chlorophyll pigments, carotenoids and phycobilins. So first we come to the chlorophyll pigments. Now, the chlorophyll pigments are the most important photosynthetic pigments that are known to occur in 7 different forms, visibly chlorophyll A, chlorophyll B, C, D and E, bacteriochlorophyll and bacterioviridin. These molecules differ in their absorption peak, empirical formula and distribution in the plant kingdom. Then we come to carotenoids. Carotenoids are lipoid compounds and they are widely distributed in the plants, and they have been reported from photosynthetic bacteria, algae and all higher plants. So they belong to a large group of compounds and they are called terpenoids. Carotenoids are of two types: carotenes and carotenols. Carotenols or xanthophyll. Carotenes are unsaturated hydrocarbons which consist of long chain of carbon atoms with 6 carbon rings at both ends, and they are of orange to yellow color and they exist in 4 isomeric forms. So they are orange to yellow color, and carotene is predominant form. Empirical formula is C40H56 and the name of carotene ends with the suffix ENE. So, carotenes. Carotenols or xanthophyll are oxygen derivatives of carotenes and they occur as alcohols or ketones. They are of yellow color. Green leaves contain more xanthophyll than carotenes. And the carotenoids, they perform 2 main functions. They prevent photooxidation of chlorophyll and they transfer light energy to chlorophyll. Therefore, they are also called as accessory pigments. Phycobilins. The chlorophyll and carotenoids are soluble in hot water and they resemble chlorophyll in having a tetrapyrrole which is open chain, and they do not have magnesium or phytol, and they absorb light in those spectra where chlorophyll shows insignificant absorption. However, phycobilins transfer the energy ultimately to chlorophyll. Okay, so these were phycobilins. Now we come to the mechanism of photosynthesis, and the overall mechanism or process of photosynthesis can be divided into 2 main phases, visibly the light reaction and the dark reaction. So now we begin with the light reaction. Light reaction includes a series of reactions which take place in the presence of light only. So, light reaction means light is a necessity. These reactions take place in the grana of the chloroplast. You have studied the structure of the chloroplast in detail. So, where is the light reaction taking place? It is taking place in the grana of the chloroplast. In this phase, light energy is utilized to produce an energy-rich compound and a hydrogen carrier. the chlorophyll molecules are in a ground state, and when light energy is there, these chlorophyll molecules get excited by the light energy and they release energy-rich electrons. Now, a smaller part of the energy of the electrons is used in converting adenosine diphosphate into adenosine triphosphate. And this conversion of ADP into ATP is called photophosphorylation and is stored for immediate supply. But the greater part is used in breaking the water molecules. This breaking of water molecules is called photolysis into oxygen and hydrogen. Oxygen is released as a byproduct, while hydrogen is picked up by the hydrogen acceptor NADP, nicotinamide adenine dinucleotide phosphate, and it becomes reduced to NADPH2. The reaction of photolysis, also called the Hill's reaction, can be represented in equation form, like protons 4H plus are used to reduce NADP to NADPH2 after they are joined by electrons which travel through a system of different electron acceptors. During this passage of electrons, ADP and inorganic phosphate, ADP plus Pi are combined to form ATP, and the process is called photophosphorylation, a term given by Arnon. Oxygen released during this process is a byproduct. Emerson reported the existence of two distinct light-driven processes. One of these was driven by wavelengths which exceeded 680 nm and the other by shorter wavelengths. These two light-driven reactions were named as photosystem 1 PS1 and photosystem 2 PS2 and respectively by Dyson and his collaborators. The photosystems or pigment systems are concerned with harvesting the energy of sunlight. Now here you can see that schematic representation of harvest of light energy by photosystem 1 in A diagram, first one A, and in B, photosystem 2 in B. So here you can see the photosystems or pigment systems, they are concerned with harvesting the energy of sunlight. Each photosystem has got a reaction center, core complex or trap, represented by a molecule of chlorophyll A, and it is intimately associated with several other molecules of chlorophyll and accessory pigments. These collectively form the light harvesting complex or antenna complex, and the energy gathered by the antenna complex is ultimately transferred to the reaction center. So here you can see that light energy is being absorbed by the various chlorophyll molecules in both the photosystem 1 and 2, and you can see the reaction center P700 in A and P680 by 90 here in B. They are in a ground state, and as soon as light reaction takes place and photons are being absorbed, the chlorophyll molecules which were in ground state, they become excited, and at the reaction center they release an electron. Photosystem 1 and photosystem 2. Its reaction center consists of a single molecule of chlorophyll A which absorbs light of 700 nm wavelength. It is designated as P700. The antenna complex is made up of several molecules of chlorophylls, phycobilins, xanthophylls, plastocyanin, cytochrome, etc. It is rich in chlorophyll A, iron and copper, and is active both in red as well as far red light. It takes part in both cyclic and non-cyclic photophosphorylation. Photosystem 2, its reaction center is a single molecule of chlorophyll A, 680 designated as P680. Its antenna complex contains a much higher number of chlorophyll B molecules than chlorophyll A, carotenoids and plastoquinone, and it absorbs light of shorter wavelengths only. It participates in photolysis and non-cyclic photophosphorylation. So you can see the difference between photosystem 1 and 2, and the main difference being that in photosystem 1 the reaction center is a single molecule of chlorophyll which absorbs light of 700 nanometers wavelength, and in photosystem 2 it is a single molecule of chlorophyll A 680 designated by P680, and it absorbs light of 680 nm. Okay, so these were the two differences between photosystem 1 and photosystem 2. In our next class, we'll study about the production of assimilatory power. Thank you.